Hi, I'm Natalia, and I'm a PhD student with Dario Farina and Juan Gallego, and I'm going to present this project about how neuromodulation produces deviations from classic motoneuron control. So essentially, to execute any kind of motor activity, we need to coordinate the activity of our joints and muscles. And to do so, the spinal cord has projections to the muscles through the alpha motor neurons. And then we can say that when one motor neuron innervates several muscle fibers, that's a motor unit. Then traditionally, motoneuron control has been defined based on Henneman's size principle that states that motor neurons are recruited from smallest to largest as they receive a common input, and also that as the force increases, the firing rate of these neurons also increments, and that's what we call rate coding. However, there are several studies that have started to prove that there might be some deviations from this constraint and rigid view. An example of this is this study in where they stimulated different areas of motor cortex through different electrodes, and then they argued that if there was read control, this stimulation would always activate the same set of motor units in the same order, and we can see that that's not what happens with the red and the blue motor unit. And in this case, they explain that this might be happening due to the presence of different common inputs to the pool. So another example is this other study in where they compare the discharge rates of pairs of motor neurons while participants perform an online control task. And in this case, they observe a prolonged firing activity that corresponded with a deviation from read control. And in this study, they hypothesized that this might be happening due to neuromodulatory inputs. So based on this last study, we believe that neuromodulation is quite important in order to produce certain deviations from read control. So in the past, it was thought that the relationship between the input and the output of the motor neurons was linear. However, what really happens is that the brainstem releases monoamides, such as serotonin or norepinephrine, that make this transfer function become nonlinear. Then with this, we can say that neuromodulation refers to any of the variations in the excitability of motor neurons that are caused by this neurotransmitter release. Moreover, the most important neuromodulatory effects in the motor neurons are the persistent inward currents, or PICs, or peaks, as they are quite important for gain control. For example, if you pay attention to this plot, we can see that a higher monoaminergic drive can produce a higher gain, and also they are quite relevant and beneficial in situations when we need a constant activation of the motor neurons, for example, when we need a proper postural control. Then the main neuromodulatory effect that we are interested in studying is the hysteresis, because that is basically a prolonged firing activity that happens even after the input has stopped. So for example, if there are no PICs, in this plot, we have the firing rate against the injected current to the motor neuron, we would have a linear trajectory. But if there are PICs, we would get this non-linear behavior that is a prolonged firing activity between the start and the end of the firing. So then the main goal of this project is to create this computational model that can replicate the intrapool variability of large populations of human motor neurons in order to study the neuromodulatory constraints for producing certain deviations from rigid control, and also to study how neuromodulatory effects can change over learning. And then to do this, we follow these steps. First, we constructed the model, then we replicated the online control experiments, and lastly, we studied the neuromodulatory effects and their influence on learning. So first, we implemented a biophysical model of a motor neuron that builds upon Hodgkin and Huxley equations, and this model receives excitatory, inhibitory, and neuromodulatory inputs and outputs the action potentials that can be converted into force. Then each motor neuron has two compartments: a dendrite that includes the excitation, the inhibition, the leakage, and the calcium channels, and also a soma that incorporates the sodium, leakage, and fast and slow potassium channels. And the way we implemented the neuromodulatory effects in the model was through the calcium channels by adding this parameter that is a voltage activation threshold of the calcium channels that basically produces a prolonged firing activity or hysteresis. So this means that the higher this value is, the more hysteresis we get, as we are lowering the threshold to fire an action potential. Additionally, to create the motor neuron pool, we just linearly interpolated the values from the smallest to the largest motor neuron. And then with this, we validated the model using experimental recordings from 8 subjects using their tibialis anterior muscles while they perform trapezoidal isometric contractions. And the most important thing about this data set is that it has a really, really high number of motor neurons per subject, around 130, and also that all these motor neurons present a really high variability between each other. So we chose a triangular contraction as the excitatory input because we were interested in the effects of the ramp up and the ramp down. And also we studied which one was the best inhibitory profile to replicate our data. So we know that an increase in inhibition should always decrease the neuromodulatory effects, in this case the hysteresis, And to study this, we run several simulations. So if we pay attention to this heat map, on the x-axis, we have different values for the inhibition. On the y-axis, values for the voltage activation thresholds of these calcium channels. And also, the darker the color is, the more hysteresis we have. And each of these squares represents an independent simulation. So knowing all this, we can see that as we increase the inhibition, there's less hysteresis. That is exactly the effect that we wanted to mimic. However, if we use other inhibitory profiles, such as the inversely proportional one or the proportional one, we didn't replicate this effect. And that's why we discarded these profiles as possible initializations for the model. We further optimize these voltage activation thresholds of the calcium channels using a black box optimizer that works like a hyperparameter sweep, in where we try to minimize the error between the simulated and the experimental hysteresis. And as you can see here on the right, the accuracy for all the subjects was quite high. Moreover, it's important to know that the neuromodulatory inputs didn't follow a clear distribution across the pool. So here on the right, we have the plot on the x-axis, we have all the subjects, and on the y-axis, these voltage activation thresholds of the calcium channels. And we can see that there's not obvious distribution across the pool. Also, we wanted to see if the model was reliable, and to assess that, we replicated different force tracking tasks using PD controllers. For example, in this case, I'm showing the case of a sinusoid in where we computed the new excitatory input in order to match the force target. So in this slide, we are just seeing a simulation example of a motor neuron pool. On the left, we have a raster plot, and on the right, the firing rates for each of the motor neurons. And the most important thing to see here is that we were able to replicate the variability in the recruitment times. And here on the bottom, we see the firing rate state space plot for a pair of motor neurons with different intrinsic properties in terms of the calcium channels and similar recruitment thresholds. And this is the case that we were interested in studying. Then, knowing this, we can say that there's rigid control when a trajectory follows a one-dimensional monotonic manifold, and any deviation from it implies a deviation from rigid control. With this, we can run simulations without and with neuromodulation, and we can observe that when the neuromodulatory inputs are present, there's an increase in firing activity, meaning that neuromodulation is sufficient to produce certain deviations from this rigid control. However, we can assess how much flexibility there's in the pool by using certain metrics. For example, we can use a displacement that calculates the difference in firing rates for two motor neurons when they vary in opposite directions. So then here, if we go to this plot, on the x-axis, we have the metric values for when there's no neuromodulation, and on the y-axis, when there's neuromodulation. With this, we see that this metric increases if the neuromodulation is present, and also if the intrinsic properties of the motor neuron pair are more distinct. So contrary to what we saw in other studies, in here we see that even if the motor neuron pool is constrained by a common input, we can observe certain deviations from classic rigid control. Then we wanted to replicate the online control experiments in where they correlated the activity of motor neuron pairs with similar recruitment thresholds. So basically, participants had to first recruit the lower threshold motor neuron, then activate both of them, and then in the last place, in order to reach this target area, they had to decorrelate the activity of the higher threshold motor neuron without activating the other one. So we implemented this in the model using a PD controller that computed two different Euclidean errors depending on the firing rates of these motor neurons in order to reach this target area. And it's important to know that we made one main assumption, that is that motor neurons receiving more neuromodulatory input had to receive less hysteresis. And this is based on previous experimental studies. Also, importantly, we could only replicate this study if the pair of motor neurons had different intrinsic properties. So then, knowing this, you can see that we could obtain a decorrelation of the motor neuron pair, even if we use different initializations, either a proportional or an inversely proportional one. In here, we have the simulated force for the online control experiments, and we can see that in order to reach the target area, they always had to decrease the force a little bit and then increase it a little bit more. That is exactly what it was observed in experimental studies. So this was a nice check. So in the last place, we wanted to study the experimental data from the online control experiments that I just explained. So it was a 15-day learning task where they had to learn how to decorrelate these motor neuron pairs, and this was performed in 6 participants using their tibialis anterior muscles. So before I give more details, it's important to know that these participants could successfully complete the task since the first day. So then knowing this, so yeah, basically knowing that here we have the force trajectories for several days. On the left, we have the force for the 2 first days, and on the right for the last 2 days. And what we see is that the force during the last days becomes smoother, which indicates a better control. However, to quantify this, we use different metrics. For example, the average number of peaks to calculate the smoothness, and also the trial duration. And we can see that it decreases across days, implying that, of course, there's a better control. So now we know that the performance is improving, but now the question is it happening due to physiological changes? Like, for example, is the inhibition changing across days, or is something like that? Or is like something more behavioral? So to analyze this, we implemented a generalized linear model, or GLM, to evaluate the correlation over days. So the idea behind this is that we have the whole motor neuron pool, then we take one motor neuron out, and then we predict the activity of that single motor neuron, either using the spikes of the rest of the neurons or the force, that are both the spikes and the force are a proxy for the common input. The only thing is that the force has less noise. So then here on the top, we have the pseudo R square for the force model, and on the bottom, the pseudo R square for the spike model. And what we observe is that the accuracy doesn't change that much over days, meaning that the common input doesn't change that much over days. And also we run the same analysis for the simulated data, and we observed exactly the same effect. The only thing is that the variability was much larger because we are also simulating a much larger pool, maybe around like 200 motor neurons. So the conclusion from this is that there's no change in the common input across days. So now it looks like, of course, there's like a behavioral change involved in this to produce a better control. So in the last place, to study this, we gave as an input to the model the force trajectories, and then we computed the spikes and also like the different firing rate state space plots for different motor neuron pairs. And what we observing here is that during the first days there was more variability. Also, we recruited many more motor neurons. That is exactly what we observed experimentally, both in the rasters and also in the firing rate state space plots. So the conclusion is that, yeah, there's a behavioral change involved in this, and that's why the control is better. So the take-home message is that we can mimic the variability observed in large pools of human motor neurons by implementing this biophysical model of a motor neuron pool, and we also optimize it with experimental data. Also, we saw that neuromodulation is necessary and sufficient to produce deviations from rigid control. And in the last place, although a neuromodulatory input is necessary to replicate these online control experiments, in order to get a better control, participants just needed like a kind of behavioral adaptation. And as a summary, is that we saw a way to produce deviations from classic and traditional and rigid control. So thank you so much, especially to Simon, Mario, Dario, and Juan, and also to you for being here. So thank you.